we're going to talk about the brain inside the skull. So, how do you see a brain? As perhaps wheels within wheels, a biologic mechanism, multiple interacting parts working automatically, or perhaps as thoughts bubbling up and then releasing like bubbles in a carbonated drink. Or maybe you see a brain like a tissue, something packed into the skull, ready to be unwrapped. Blood vessels, proteins, uh, a mass of biology. Maybe you see it more schematically, like this colored coded section of the human brain, the frontal lobe, the occipital lobe, and the temporal lobe, parietal lobe. We're going to be talking a lot about the occipital lobe, of course, but all the whole brain is going to come into play. So we left the eye last time, and we know that the ganglion cells made up cell processes that became fibers of the optic nerve in behind the eye in the orbit. Here is a picture of the orbit, and the small yellow arrow is pointing to the hole that the optic nerve uses to leave the orbit and enter the skull. Now, how does it become part of that brain within the skull? How do the images travel? Well, first, let's look at how the images are organized. We see here a very simplified cartoon. We have the visual field as blue on one side, green on the other side. We have overlapping fields where we'd have the best stereo vision. And we see that images on one side of the visual field enter the eyes. So if it's off to my right, it enters the eyes towards the left. The nasal on my right eye, temporally the outside on my left eye. These combine, they cross at something called the chiasm and then segregate into the left visual field and the right visual field at the level of the lateral geniculate nucleus, the LGN. The lateral geniculate nucleus, we're not going to get into great detail about it, though it's fascinating. It's sort of pyramid shaped. It has layers, and the fibers go to specific layers in part from specific types of ganglion cells. Here we see the cross section of the lateral geniculate nucleus circled in the blue circles on both sides, with now the corresponding visual fields again color coded here. But there's an important point. The LGN gets only about a fifth of its impulses from the optic nerve, from the retina. Most of its impulses come from different parts of the brain and modulate the functions of the LGN. This is critical. This is completely new. We didn't see anything coming from outside the eye other than photons of light. This is now the brain doing what brains do. It's complex. It modulates. And it could even be modified, some say, by mental states, coloring our vision, as it were. This is one of the places that happens, but it happens on many levels. So after the lateral geniculate nucleus, where does it go? Well, it goes to the back of the brain, the visual cortex, also called in the specific area that it first goes to is V1, visual area 1. Kind of makes sense. Many of these visual areas, that are usually classified as about 6 of them, have other names, but we won't bother much with that. Now, the positions of the visual scene that we saw in the retina and the visual fields are still segregated here. We still have a scene that we transmitted from the retina, but it is, just like in the retina, heavily edited. Well, not so much edited, but selected for central vision. Half of this V1 area is dedicated to 1% of the vision, the foveal vision. So we see right away the brain is saying, this is what's important. We're going to agree with the eye. We're going to agree with what you're looking at. Now, there's also a special love for lines here. In areas of this visual cortex, as you go from one part to the adjacent part to the adjacent part, there are orientations of lines that are favored, from vertical to horizontal. And to go 180 degrees could be a full millimeter of this cortex, which is pretty far by brain standards. There's a lot of cells. The right and left eyes are in adjacent areas too, and there's also some framing here now for brightness and movement. Now movement is being captured. There's some indications that you see some evidence of movement in the eye, but how would you stitch them together? There's this scene, then something moves, the scene changes. The eye can't deal with that alone. Here we're starting to deal with that. We're starting to favor movement. Now, after this primary cortex is V1, we go, up the hierarchy to V2. This responds more to contours, textures, the location, something as far as figure and ground, what's in the foreground, what's in the background, starts to be parsed out here. There's the next big one really is V4, which is tuned now again towards orientation, but also more for color. It's tuned for more intermediate complexity. Now we're talking about simple geometric shapes. We're also starting to see salience, what's important. Now we already see that built in a bit with the foveal vision being privileged, but salience for the images and attention as well starts showing up here. Then there's multiple areas. We're back to this schematic, and we see, leaving the occipital cortex, how some people feel it goes into 2 different directions. What they call a dorsal train or pathway. That's the blue arrow going up. And the blue pathway, the dorsal pathway it's called, is supposed to be where things are. And the what pathway that figures out what it is that's where is the yellow arrow going towards the temporal lobe. Now, not all agree. Some have suggested that this stream that goes to the temporal lobe is real visual perception of things, and the dorsal stream has more to do with control of skilled actions, because that's near the motor cortex and entails it. But you get the idea. There are these basic pathways that go up with multiple functions. Now, in this what hierarchy, what it is, one of the major areas is this inferior temporal cortex, the fusiform face area in the temporal lobe. Now, this is fascinating. it got a lot of press and a lot of scientific attention of this is where we see faces, and you could see how faces are built up by stimulating different areas. Is that true? Yes and no. I mean, yes, it does see faces, and that's very important, but it doesn't just see faces. Now, for example, in infants, faces are very important, and yet it doesn't seem that in studies this area is quite so important. Uh, it seems that it can be activated, but not necessarily specifically for faces. Maybe that's learned or develops later. But other familiar things will light up that area. In adults, uh, even Chinese characters will light up that area. And I've had a thought about that because I've noticed that I seem to process very familiar groups of hieroglyphic, Egyptian hieroglyphic signs differently than trying to sound them out, which you can do because I know what they're supposed to say and how we transliterate them. But I don't have to, they're so familiar. I've seen them on so many monuments and so many texts that it just, I see that pattern and I know what it means. There are these very standardized patterns. Uh, Now, also, even blind people will light up that area when feeling a face. There you can argue, okay, well, it's still if a facial area, but again, it seems to have more to do with salience, familiarity, need to know. So yes for faces, and no, maybe calling it a face area is a little misleading. Now, we talked in the lateral geniculate nucleus, the LGN, that only 80% of the inputs were from other parts of the brain. Here we see a cartoon showing how different areas are feeding into each other, particularly different sensory areas. I mean, when we put together a scene, when we want to check it for what it really is, we will incorporate everything. We smell, especially when we taste, but we also will look at the color. Say if you're into wine tasting, they all come into play. When we see a bird, we're listening for its sounds. We're not only just looking at it. We check in with memories. We have to put it together and interpret it. So we use a lot of information in the brain to see a scene and to have it make sense to us. Now, you may have heard the rumor, the wishful thinking, I think, that we only use 10% of our brain. And what I mean by wishful thinking, I think people want that to be, oh, we have 90% of brain. We could be so brilliant and the world would be better if we just, no. That's clearly not how this works. Look, you're using your whole brain the moment you look at it, not your whole brain. Well, some people think it's close. You're using 50%, they feel, for visual processing. Some people will say closer to 70, 80, 90%. I guess it may also depend what you're looking at, the complexity of it, and what other senses are engaged, how much memory you're investing in this, those kinds of things. But you're not just using 10% of your brain. It makes no biological sense. We don't waste expensive tissue like this. Brains take a lot to develop, and they use a lot of oxygen, a lot of blood flow. We're not just keeping it up there so maybe you can learn some special technique to unlock the secret. We use our brains. We may or may not use them well, but we use them. So some other examples of what the brain does with vision. Remember that we said that the visual image on the retina is inverted, left, right, up, down? Well, there were experiments first actually done by the scientist Helmholtz, who was very interested in vision, in 1861, then an American professor in 1896, but more notably by the Austrian professor Theodor Erisman and his assistant Ivo Kola in the mid 20th century. More notable because they made a film of it, and they really kind of played it up. They found that in these experiments, if you wore prism glasses or other ways to turn your world upside down, really goggles, and wore them all the time, you can get used to it in a week. That's right. You no longer experience it as an upside down world. By 10 days, they were riding bicycles, and later they did some work with left to right inversion, and they found one of them could ride a motorcycle in town in just a few weeks. And I guess didn't hurt themselves or anybody else. An important point. This learning is not an intellectual effort. You can't make it happen. You can only experience it. You can only let it develop. You can't think your way through this. You can't willpower it and bully it into changing. You just have to experience it. You just have to live it. Interestingly, you can buy these glasses or goggles. They're sold in part as a party favor for kids, so they can, see what it's like to be in a different world. Others sort of upsell it as experiencing a new reality, or maybe for training your brain if you have motion sickness or what have you. They're not hard to get. They're still out there. Now, one of the things about this is the brain isn't privileging an orientation here. It doesn't care how the image is. It's flexible. It's contingent. It is open and will work with what you give it, within some parameters, of course. This is really adaptation. It's not fixed. It's not something that is, again, a bug. It's more of a feature. Now, some clinical examples. There's amblyopia. Amblyopia, also called lazy eye, for probably a couple of different reasons. One reason is because strabismus and eye turning in and out may be a cause of it. Also because the eye seems lazy. Even if it looks normal, it's not seeing. Now, this develops early in life when one eye sees more poorly than the other. Say you're farsighted, so as a baby you're not seeing your toys and food that are up close, so you just give up if you're farsighted in one eye and nearsighted in the other, you're going to give up the more farsighted eye because you're not driving a car or watching TV as an infant. Of course, it could be due to an injury, or a congenital cataract, or a cataract from an injury that develops when you're young. For example, when babies have congenital cataracts, it's actually more of an emergency when only one eye is involved, because that eye is going to get amblyopic in days. It's very fast in infants. It could take weeks to months in older children, but it can happen in days in an infant. So they actually do surgery right away, or as soon as conceivably possible. The whole point is, just like we said before, brains don't waste energy. Biology is not efficient if it's using unneeded resources. So if the brain doesn't see a clear image from one eye and it does from the other, it's going to say, we know what we're going to invest in as a brain. Now, you can reverse it by forcing the bad eye to work, but that's only going to work and be effective if the bad eye has some potential vision. Now, sometimes if there's a cataract, if there's blood in the eye, if there's a strabismus that you fix surgically, or you can give glasses, then the eye has a chance. Now, this is only really well demonstrated till about 7 years old to be clearly effective. You might patch the eye. Sometimes they use eye drops. But the idea, though, is that after 7, you can still get some gains, but it's not going to be as good. The brain is more plastic early in life. The sooner you get to it, the better. Now, I want to give another example, one that I've personally experienced, is migraine scotoma, scintillating scotoma, fortification spectra. There's a lot of names for them. I found that interesting because I got them without the actual headache, but it's still a migraine phenomenon. It's a vascular phenomenon of spasm dilation. And you get this scintillating, vibrating vision that starts centrally and develops. A lot of times you just notice in one eye, but if you really notice, it's less in the eye with the nasal visual field because that's smaller. It's most often both sides because you don't get patterns like that really in the retina. There are probably really such a thing as retinal migraines, but most of these are in the brain. And you project it out there. You don't see it as in your brain. You're seeing lights out there that you know aren't there. Um, Now, what about the building a world in for blind people? There really is some truth to the stories that there are more enhanced connections uh, for hearing, smell, touch, and even memory when somebody has gone blind early in life. Remember, you don't let real estate, you don't let the fields of the brain go fallow. You use them. The brain seems to really rewire uh, outside the visual cortex to expand its world. Uh, there are studies that show those who are blind, either from birth or very early in life, do truly have better hearing when you do different types of hearing tests. Not just in how much they hear or how loud, but I mean in terms of quality and interpretation. Brains of blind people reading with Braille, they light up their same areas that sighted people do when they read a book or a tablet. If blind later in life, it's different. You still may be more sensitive to sound and other senses, smell and all that, because you pay better attention to them. Uh, and your brain does seem to redirect some of its abilities of vision to these other areas. So indeed, uh, we see that even people who go blind later in life will show some evidence of being able to use these other senses more effectively. There's also a syndrome for those who go blind later in life where they will hallucinate, sometimes formed hallucinations, sometimes not. And it can be very tricky. This is called Charles Bonnet syndrome. I mean, the key here is brains want to brain. Visual cortexes want to visually cortex. And it can be formed hallucinations. It can be very frightening. Sometimes it's just so obvious that the person is not as upset as you might think. It's usually with more profound blindness. Now, there's another phenomenon that I want to talk about, and this is echolocation. Blind people will actually make sounds and echolocate, and it can be incredibly effective. You can look for videos about this. They can ride bicycles, and there was a lot of skepticism about it, but I think a lot of that skepticism has dissolved. I actually saw a presentation at Jules Stein at the Eye Institute at UCLA at a grand rounds maybe 15 years ago, and it was really quite impressive. And you can even play with that for yourself, whether you're sighted or not. Just make sound, make pings, and see how it echoes. It seems to be very effective. And again, it's clearly us being sensitive to our environment and using other senses to build that scene, to build that world. So, what have we seen? We've seen that the brain is much more complex than the eye. It has much more inputs, and the wheels within wheels are huge and brain wide and entail many functions, different senses, memory, salience. And it's also much more flexible than the eye. The mechanics are much more complex, and yet they have plasticity. You may have heard that term, of course. The brain does have plasticity. But like the eye, it also builds an image with the heuristics of salience, what's important, and with a huge interplay between parts of the scene. One of the things that blows my mind is how fast this happens. I mean, close your eyes, look to the side, open your eyes, and you say, well, I know it's there. No, just look. It's like this. It's much less than a second before you've registered and you know it's there. You know you're seeing it. You're not just remembering it. It may be faster with things you know and memorize. If it's a very ambiguous scene, that's different. But when you're in a car sometimes, somebody else driving, and it turns a corner that you're not familiar with, just before it turns that corner, close your eyes and open your eyes, and you'll see how much you register immediately. This blows me away how fast this happens, because remember, all of that's going on. It has to go from the eye, then within the eye it goes through several layers of cells that are all interacting, setting out their neurotransmitters, and with signals going up the neurons in the cells into the LGN, where it again has to synapse and release chemicals, and then this signal has to travel up into the V1, then 2, 3, 4, 5, 6, the temporal area, and then have all this feedback from multiple areas and multiple senses. And less than a second, unless it's, again, a really ambiguous scene, you know what's going on. And sometimes, of course, artists or filmmakers will do that. They'll purposely make a scene ambiguous until you focus in on something. I kind of find that annoying. It's actually. So, and we see the brain as a sense organ, as we talked about in the first talk, like the Buddhist formulation. It's layered, where the brain is seeing what another part of the brain saw and uses that to put together a scene. And then stitching scenes together from the visual fields of both eyes, from motion over time. After all, there's movement, there's things change, lights, shadows. It's all relationships and change. Relationships and change. Wheels within wheels. So it all happens in here, right? The phantoms we project, the chimera we create, the world we put together. Or does it? In the Shurangama Sutra that we referred to before, the Buddha said, okay, yeah, it's not the eye, we got that, or at least not the eye alone. That's perhaps necessary but not sufficient. Is it the brain? Yes and no. After all, take out a brain, an eye is not seeing, even if it's a healthy eye. But it's contingent. It's a fluid web of interactions. And then you build a scene, but where's the scene? There's no screen. There's no monitor. Who's doing the scene? Where's the screen? Who's seeing what? What's seeing what? Perhaps what the Buddha is asking is not so much about brain functions when he asks, is vision in the brain, but about where the mind is. And maybe we should too. But before we explore that further, I want to give us a little more taste, a little flavor of what's going on in the world of vision and in the world around us. And let's explore that with color. Color is full of insights. Does color even exist? What did the old Zen master mean when saying, name the color, blind the eye? Let's look at that in our next talk.